Welcome to this lecture on the fundamentals of genetics. Today, we're going to explore one of the most fascinating and fundamental processes in human biology, sex determination. We will uncover exactly how nature decides whether a developing embryo will grow into a biological male or a biological female, setting the stage for an individual's entire physical development. To begin our journey, imagine you are building a highly complex robot and you have two different instruction manuals to choose from for the final design. One manual tells the factory to build a robot with a specific set of tools, while the other provides a completely different set. In human biology, this is exactly what happens when a baby is conceived. But instead of paper manuals, we use microscopic structures called chromosomes. Every time a new life begins, a biological coin flip determines which set of instructions the baby will receive from its parents. Now that we have our instruction manual analogy, let us look at the actual biological structures. In formal genetic terms, human sex determination is governed by the XX and XY chromosomal system. Humans have 46 chromosomes in total, arranged in 23 pairs inside almost every cell. The first 22 pairs are autosomes, identical in both males and females. But the 23rd pair consists of the sex chromosomes. A biological female possesses two X chromosomes, while a biological male possesses one X and one Y chromosome. Understanding the chromosomes leads us to the true master switch in this system, a specific gene located on the Y chromosome called the SRY gene, which stands for sex-determining region Y. When the SRY gene is present, it triggers a cascade of chemical signals that tell the embryo to develop male reproductive organs. If the SRY gene is absent, which is the default state when only X chromosomes are present, the embryo naturally develops female reproductive organs. Before we move into the mathematics of inheritance, we must understand why chromosomal sex determination matters so much. In basic biology, it explains the fundamental mechanism of sexual reproduction and how genetic diversity is maintained, allowing our species to adapt over generations. In modern medicine, this knowledge is absolutely crucial. It helps doctors diagnose and treat sex chromosome aneuploidies, like Turner or Klinefelter syndrome, where a person has an abnormal number of sex chromosomes. It also provides vital insights into rare cases where physical appearance does not match chromosomal makeup. With the biological foundation set, let us look at how sex is passed down mathematically using probability. The mother always provides an X chromosome because she has nothing else to give. The father's sperm cells have a 50% chance of carrying an X and a 50% chance of carrying a Y. This brings us to our first formula. P of C represents the total probability space for the child's sex. XM is the mother's X chromosome, a certainty. XF and YF represent the father's possible contributions. The fraction 1/2 shows the father's 50% chance to pass on either chromosome. If the frequency of Y-bearing sperm were to increase, the probability of a male child would increase proportionally. Now that we have our formula, let us apply it to a very common question. What is the exact probability that a couple's first child will be a biological female? We know the mother definitely contributes an X chromosome, so we only need to look at the father's contribution. He produces millions of sperm, half with X and half with Y. We set the probability of the child being female, P of F, equal to the probability that the father contributes his X chromosome. Since he has one X and one Y, the chance is exactly 1 out of 2. Converting this fraction gives us 0.50, or a 50% chance. The biological coin flip is perfectly even. To visualize the calculation we just performed, let us look at a Punnett square. On the left vertical axis, we see the mother's genetic contribution. She is XX, so she can only pass on an X chromosome, shown by the 2 rows. On the top horizontal axis, we see the father's contribution. He is XY, so he can pass on either an X or a Y chromosome, shown by the 2 columns. When we trace the arrows into the 4 boxes, we see 2 boxes result in XX females and 2 boxes result in XY males. This diagram perfectly illustrates why there is always a 50-50 chance for either sex. Having seen the probability for a single pregnancy, let us explore what happens across multiple pregnancies. To find the probability of multiple children having the same sex, we use the joint probability formula. P of T represents the total combined probability of having 3 female children consecutively. The terms P of F1, P of F2, and P of F3 represent the individual probabilities of the first, second, and third child being female. Because each pregnancy is a completely independent event, we must multiply their individual probabilities together to find the combined probability for the entire sequence. Let us put this joint probability formula into practice with a real-world scenario. Suppose a couple already has 2 daughters, and they are expecting a third child. They want to know the probability of having 3 daughters in a row. Many people mistakenly believe that after having 2 girls, a boy is somehow due to arrive. But genetics does not have a memory. We must calculate the joint probability of 3 independent events happening sequentially. We multiply 1/2 by 1/2 by 1/2. The final result is 1/8, which equals 0.125, or 12.5%. Now that we have calculated the result, let us break down the mathematical reasoning behind it. Why do we multiply these fractions? This chunk of our proof establishes the rule of independent events. Because the mother's contribution is constant, the father's genetic contribution acts as the independent variable that completely dictates the outcome. The outcome of the first pregnancy physically cannot influence the sperm that fertilizes the egg in the second or third pregnancy. Therefore, to find the probability of this specific sequence occurring, we multiply the individual probabilities together, giving us 1/8. Continuing our logical breakdown, we interpret what this 12.5% actually means. This brings us to our final result. The 12.5% chance only applies to the entire sequence of 3 births, viewed as a single combined event from the very beginning, before the couple even started having children. However, for the third pregnancy itself, the chance of having a girl is still exactly 50%. The biological coin flip always resets. Understanding this distinction proves why previous children do not change the fundamental 50-50 odds of the next pregnancy. To wrap up our lecture, let us review the key takeaways. Human sex determination relies entirely on the XX and XY chromosome system, with the SRY gene acting as the master switch for male development. Because the father can pass on either an X or a Y chromosome with equal likelihood, he is the genetic parent who ultimately determines the biological sex of the child. Finally, every single pregnancy carries an independent 50% chance of resulting in a male or female, ensuring a roughly equal balance of sexes in the human population. You now have a solid understanding of the fundamentals of human sex determination.